Welcome student in Christchurch Convent Digital Classes, Class 12, Subject Chemistry, Chapter 3, Part 3. In this video, we'll see the function of salt bridge in detail and its composition. So, as you have seen in the Daniel cell and the previous video, this is our salt bridge. The red spot moving in the video is a salt bridge. Now we'll see how it is made up of. Salt bridge is an inverted U-tube like structure, as you see in the diagram. It is an inverted U-tube like structure. The tube is filled with concentrated solution of an inert electrolyte. Electrolytes are nothing but salt like KCl, KNO3, NH4NO3, ammonium nitrate. So these are salts or compound used as electrolyte. The electrolyte or salt does not undergo any sort of chemical reaction, nor does it react with concentrated solution in the two half cells, that is CuSO4 and ZnSO4, means zinc sulfate and copper sulfate. Most of the time the salts like KCl, KNO3, NH4NO3 are used as electrolyte. This is filled, the solution of these salt are filled in inside the U-tube glass, inverted U-tube glass vessels, which is sealed at both ends with the help of glass wool. Salt bridge is constructed by filling the U-tube with a mixture prepared by mixing agar. It is a gum obtained from plant, or gelatin. Gelatin is a fat-like substance obtained from animals. Capsule cell are also made up of gelatin. These are two types: hard gelatin capsule and soft gelatin capsule. Pudina rice comes in soft gelatin capsule, and Neurobion Fort capsule have hard gelatin packing with a hot concentrated solution of the electrolyte. The apparatus is then cooled and consequently the solution fixed with the U-tube in the form of gel. So a gel-like substance is filled inside the salt bridge. Now this gel-like substance helps or prevent the intermixing of fluid of zinc sulfate and copper sulfate. The either end of the tubes are then sealed with using cotton wool or glass wool in order to reduce diffusion. The main advantage of using this bridge or salt bridge is that it prevents the arousal of potential difference, also called liquid junction potential, when two solutions are in contact with each other. Which two solutions? Zinc sulfate and copper sulfate. What is potential difference? It is the difference in the voltage between 2 points in bringing unit positive charge from infinite to those two particular points. It also completes the electrical circuit as it connects the electrolyte in the two half cell. So in board examination, function of salt bridge is asked in 2 marks, 3 marks. So you can write these points and you will get the marks. So these are important. Please learn them by heart. It avoids the chances of diffusion of solution between the half cells, means the beaker containing zinc sulfate and copper sulfate. Salt bridge also maintains electrical neutrality by providing the path for the movement of ions. So these are the main functions of salt bridge. You must remember them. Sometimes question is asked in examination, so these are very important. You can learn these point: first, second, third, fourth. So in this slide you have four functions of salt bridge. You can make notes also. Now we will see how the measurement of electrode potential in a cell is done. For this, we take a half cell of standard hydrogen electrode, which is in short it is called SHE, standard hydrogen electrode. In standard hydrogen electrode, which act as an anode in this particular combination with copper sulfate solution and copper electrode dipped in it in another beaker, you can see here. So oxidation take place. This is the tungsten filament, and it is covered with this is tungsten metal wire, and it is covered with U tube, inverted test tube like glass jacket, which is opened at one end, from where hydrogen gas is sent inside the glass tube at 1 bar pressure, and the solution inside the standard hydrogen electrode, whose concentration is 1 molar hydrogen ion concentration at 25 degrees centigrade at 1 atm. So working and construction of a standard hydrogen electrode is also asked in examination, where in this particular case oxidation is taking place. Now another half cell is in another beaker containing copper sulfate solution, and copper cathode is there dipped inside the solution, and reduction is taking place here, and these two anode and cathode are connected with the help of a wire, metallic wire, and voltmeter is also connected. We have connected voltmeter so that we can measure electrode potential of a cell. The anode, made up of platinum metal or electrode, is connected to standard hydrogen electrode SHE. Here copper electrode, which act as a cathode, deposit dipped in copper sulfate solution, is connected with a standard hydrogen electrode made up of platinum anode covered with inverted test tube like structure, which is open at one end, from where hydrogen gas is sent inside the 1 molar hydrogen ion solution at 1 atm at 25 degrees centigrade. So standard hydrogen electrode and copper cathode, both these two form a cell, of which we have to find the potential difference. The electrode forming negative terminal of the cell is allotted to negative value of electrode potential, whereas the electrode forming the positive terminal of the cell is allotted to positive value electrode potential. The potential difference developed between the two terminal is measured with using a potentiometer. You will also study about potentiometer in physics also. And what is the use of potentiometer or voltmeter? It is measure voltage. or potential difference between 2 points of electric circuit. The direction of the flow of current in the external circuit is identified using galvanometer. Galvanometer shows the direction of current. What is the use of galvanometer? Galvanometer shows the direction of flow of current. Thank you for watching video. We will see further in detail in the next video.